Bismillahirrahmanirrahim. The three major components of the body's immune system are B lymphocytes or B cells, T lymphocytes or T cells, and the innate immune system. B lymphocytes are mainly derived from bone marrow cells, while T lymphocytes originate from the thymus. The B lymphocytes are responsible for the synthesis of circulating humoral antibodies, also known as immunoglobulins. The T lymphocytes are involved in a variety of important cell-mediated immunologic responses such as graft rejection, hypersensitivity reactions, and defense against malignant cells and viruses. B and T cells respond in an adaptive manner, developing a targeted response for each invader concerned. The innate immune system defends against infection in a non-specific manner. It contains a variety of cells such as phagocytes, neutrophils, natural killer cells, and others. Immunoglobulins are composed of multiple polypeptide chains. These are oligomeric proteins whose individual subunits are classified as heavy and light chains based on their migration during gel electrophoresis. Here the heavy chains are shown by the orange color and the light chains by the yellow color. Human immunoglobulins can be grouped into 5 classes abbreviated as IgA, IgD, IgE, IgG, and IgM. The basic tetrameric structure of immunoglobulins consists of 2 identical light chains or L chains and 2 identical heavy chains or H chains. The heavy and light chains are synthesized as separate polypeptides, glycosylated to form glycoproteins, and assembled within B cells or plasma cells into mature immunoglobulins. The immunoglobulin IgG has basic tetrameric structure only, whereas IgA has 2 copies of the basic tetrameric structure. IgM has 5 copies of the core tetrameric structure. The type of H chain determines the class and the effector function of the immunoglobulins. Alpha chain is for IgA, delta chain for IgD, epsilon chain IgG, gamma chain epsilon chain IgE, gamma chain IgG, and mu chain for IgM. The 5 types of H chains are distinguished by differences in their CH regions or their constant region. C stands for constant. The gamma chains of IgG are organized into 4 conserved domains: an amino terminal variable region or VH, and 3 constant regions abbreviated as CH1, CH2, CH3. The mu and epsilon chains each have 4 CH domains instead of the usual 3. The constant regions determine the class-specific effector functions. The constant regions of the immunoglobulin molecules, particularly the CH2, CH3, and CH4 of IgM and IgE, located within the Fc fragment, are responsible for the class-specific functions of the different immunoglobulins. There are 2 general types of light chains as shown by yellow color here, kappa and lambda, which can be distinguished on the basis of structural differences in their CL regions. A given immunoglobulin molecule always contains 2 kappa or 2 lambda chains, never a mixture of kappa and lambda. In humans, kappa chains are more common than lambda chains in the immunoglobulin molecules. The site on the antigen to which an antibody binds is termed antigenic determinant or epitope. The region between the CH1 and CH2 domains is the hinge region, shown by dull black color here. The hinge region confers flexibility and allows both Fab arms to move independently. This facilitates binding to antigenic sites that may be variable distances apart or even on different bacteria and viruses. Thus, antigen antibody clusters may be formed, which can be easily spotted and cleared by the macrophages. As antigen binding site is formed when the hypervariable regions of H and L chains align together in three-dimensional space as projecting loops from the antibody surface touch the structure. The antigen antibody interactions take place because of the mutual complementarity between the surfaces of the CDRs and epitopes that involve multiple non-covalent interactions such as hydrogen bonding, salt bridges, hydrophobic interactions, and van der Waals forces. The variable regions of the immunoglobulin light and heavy chains form antigen binding sites, leading to specificity of antibodies for antigens. These are heterogeneous to the extent that no two variable regions from different humans share identical amino acid sequences. The variable regions of the L and H chains contain few or around 5 to 10 residue islands called hypervariable regions. These are also called complementarity determining regions or CDRs, shown in blue color here. Various combinations of H and L chain CDRs can give rise to multiple antibodies with wide range of specificities. This feature is called combinatorial diversity. Large antigens interact with all the CDRs of an antibody, whereas small ligands may interact with only one or a few CDRs that form a pocket or groove in the antibody molecule. Approximate percentage wise distribution in serum for IgG is 75, IgA 15, IgM 9, IgD 0.2 and IgE 0.004. Approximate serum concentration in milligrams per deciliter for IgG is highest around 1000, IgA 200, IgM 120, IgD 3 and IgE 0.004. IgE 0.05. Sedimentation coefficient, which is a quality related to the size of a microscopic particle equal to the terminal outward velocity of the particle when centrifuged in a fluid medium divided by the centrifugal force acting on it, expressed in units of time. For IgG is 75, IgA 75 or 115 depending upon whether it is monomeric or dimeric form, IgM 195, IgD 75 and IgE 85. Molecular weight of IgM is highest. IgG, IgD and IgE are present as monomers. IgA may be present as monomer or dimer, whereas IgM may be in monomeric or pentameric form. IgG and IgM fixed complement. Transplacental passage is documented for IgG. IgE is involved in mediation of allergic response. IgA is found in secretions. Opsonization is the coating of a particle with protein that facilitates phagocytosis of the particle by tissue macrophages. IgG plays part in opsonization, whereas IgM forms antigen receptor on B cell. A J chain is a protein component of the antibodies IgM and IgA. It It is a 137 residue polypeptide. So IgG is mainly a secretory antibody. It opsonizes the bacteria and fixes the complement. IgA is a secretory antibody, prevents attachment of bacteria and viruses to the mucous membranes. IgM is produced in the primary response to an antigen and it also fixes the complement. IgD acts as a receptor for antigen and IgE mediates allergic response. Somatic mutations are the mutations that occur in a differentiated cell rather than a germline cell.